Approximately 5,000 years ago, a mysterious disease devastated a region in China. Even today, archaeologists are finding houses literally stuffed with skeletons. This infection was fatal across all age groups, spreading so quickly that there wasn't enough time to bury the deceased. This excavation site is known as Hamin Mengha. In 430 BC, the Athenian plague resulted in the deaths of 100,000 individuals, surpassing the casualties of the Athens-Sparta War. During the Roman Empire, the Antonine Plague claimed 5 million lives. Centuries later, the plague of Justinian spread throughout the known world, and the Black Death decimated 200 million people, which was 60% of the global population at that time. In the 16th century, Europeans introduced the plague to North America, causing the death of 90% of the indigenous population and the collapse of the Inca and Aztec civilizations. The Great Plague of London killed 100,000 people and contributed to the Great Fire of London. In the late 18th century, a plague outbreak in Russia claimed 100,000 lives in Moscow. The 19th century saw a cholera pandemic with millions of victims. Throughout history, smallpox has caused 500 million deaths, while the Spanish flu claimed 100 million lives. In the late 1950s, a pandemic in China emerged from a new virus combining several existing bird flu strains, resulting in over 1 million deaths. Since the 1980s, the HIV epidemic has claimed 35 million lives. Following this, there were outbreaks of swine flu and Ebola in West Africa. The COVID-19 pandemic has also claimed 7 million lives. These devastating events were caused by viruses, some of the most enigmatic, frightening, and destructive forms of matter. In caves, human lungs, Siberian ice, the ocean, and high-altitude winds, scientists continue to uncover new viruses. They find so many that they can barely keep up with naming them. While a few thousand viruses have official scientific names, the true number could reach into the trillions. Virology is still a nascent field, and our knowledge of viruses, which have existed for eons, has largely been shaped by their effects, diseases, and death. Until recently, we lacked a clear understanding of the causes behind sudden illness, intense suffering, and severe health declines. The term virus itself embodies a paradox. Derived from Latin, where it originally referred to both snake venom and seminal fluid, representing creation and destruction, the word has evolved over time. In later centuries, it came to signify something contagious and disease-causing. This evolution in meaning reflects the true nature of viruses. It wasn't until the late 19th century that the term virus gained its modern connotation, thanks to an agricultural crisis. A disease afflicted tobacco crops in the Netherlands, causing the plants to wither and their leaves to display a mosaic pattern of live and dead patches. Entire farms were ruined. In 1879, Dutch farmers turned to young agrochemist Adolf Meyer for assistance. Meyer named the affliction tobacco mosaic. To identify its cause, he examined various factors including soil, temperature, light, environment, and the plants themselves, but found no differences between healthy and diseased plants. Meyer hypothesized that an unseen infection might be responsible and began searching for fungi or parasitic worms on the tobacco plants, but found none. He then extracted sap from the diseased plants and injected it into healthy ones, which soon became infected. Meyer realized there was a pathogen breeding in the tobacco seedlings, a microorganism capable of causing disease. Meyer takes the sap of the diseased plants and puts it in an incubator in his lab. Before his eyes, colonies of bacteria appear. Meyer applied these bacteria to healthy seedlings to see if they would cause tobacco mosaic. However, nothing, absolutely nothing happens. Few years later, another Dutch scientist, Martinus Vagrancy, wondered if something other than worms, fungi, or bacteria could cause disease, something much smaller. He pulverized diseased plants and ran the mass through a filter. The output was a clear, cell-free liquid. When Vagrancy administered it to healthy plants, they became sick with the very same tobacco mosaic. In 1898, Vagrancy described his discovery as an infectious living fluid and spreads disease. This substance, Vagrancy hypothesized, was alive, but it was different from all life forms known to him. Already at the end of the 19th century, scientists had no doubt that all living things were made up of cells, but there were no cells in his fluid. The substance that Vagrancy identified was both remarkably persistent and extraordinarily fine. He named this enigmatic agent virus, borrowing the term from the ancient Romans. Despite extensive experimentation, Vagrancy was unable to determine exactly what he had discovered. It was neither an animal, plant, fungus, nor bacterium, something entirely different. In the early 20th century, scientists using Vagrancy's methods of filtering and contamination identified additional viruses, each causing various diseases. Scientists eventually learned to cultivate some viruses outside their natural hosts, but the true nature of viruses remained elusive. Opinions varied widely. Some viewed viruses as parasites exploiting host cells, while others considered them mere chemicals. The debate continued over whether viruses were living or non-living. In 1923, British virologist Frederick Twort remarked, "The nature of viruses is impossible to ascertain." In the 1920s, chemist Wendell Stanley, then a student, mastered the technique of arranging molecules into repeating structures to form crystals. This crystalline state allowed scientists to investigate molecular structures in ways that were otherwise impossible. By bombarding these crystals with X-rays, which diffracted off the atoms and imprinted repeating patterns on photographic plates, scientists could deduce the structures of the molecules within the crystals. During this period, the use of crystals revealed that enzymes, which were known to cleave other molecules, were composed of proteins. This breakthrough led Stanley to speculate about the composition of viruses. If enzymes were proteins, might viruses be composed of proteins as well? To investigate further, Stanley turned viruses into crystals, choosing the tobacco mosaic virus for his study. He extracted sap from diseased tobacco plants and filtered it, just as Vagrancy had done 40 years earlier. After removing minute impurities, Stanley prepared the liquid for crystallization. To his astonishment, microscopic needle-like structures and films began to form, making the virus visible to the naked eye for the first time. Stanley found that the virus crystals were as solid as minerals and could be stored for months like table salt. When dissolved in water, these crystals returned to their infectious, living state, capable of contaminating everything they touched. Stanley's 1935 publication of these results astonished the scientific community, with the New York Times declaring, "The old distinctions between animate and inanimate are completely irrelevant." Despite its groundbreaking nature, Stanley's research had a significant oversight. Tobacco mosaic viruses were not composed solely of protein. In 1936, British scientists Norman Peary and Frederick Bowden discovered that 5% of the virus's mass was a mysterious thread-like molecule, later identified as nucleic acid. This substance, now known to store genetic information, is crucial to understanding viruses. Genes are stored in our cells as double-stranded molecules of nucleic acid, DNA. However, some viruses, including the tobacco mosaic virus, contain single-stranded ribonucleic acid, RNA. Other viruses may carry double-stranded RNA or single-stranded DNA. It took scientists a decade to unravel how viruses utilize this genetic material to replicate and produce new viruses. To observe individual viruses, scientists required a new generation of microscopes that use electron beams to study extremely small objects. In 1939, Kasia Gustav and Helmut Ruska added crystals of tobacco mosaic virus to drops of distilled water and placed them under the new microscope. They were able to see tiny rod-shaped structures, each about 300 nanometers long, an unprecedented view of such minuscule living entities. To grasp the scale of viruses, consider a single grain of salt on a table. You could fit 100 bacteria along one side of the cube of salt, and up to 1,000 viruses along that same edge. Over the next decade, virologists continued to explore viruses and map their molecular structure. While viruses, like our cells, contain proteins and nucleic acids, they utilize these molecules in a markedly different way. Human cells are filled with millions of diverse molecules that are in constant motion, interacting and binding together, allowing cells to perceive their surroundings, move, absorb nutrients, grow, divide, or even undergo programmed cell death. In contrast, viruses are much simpler. Typically, they consist of just a protein shell encasing a few genes. Despite their minimal genetic instructions, viruses manage to replicate by hijacking the cells of other organisms. A virus injects its genetic material and proteins into a host cell, reprogramming it to produce new viral particles. Thus, one virus can eventually lead to the emergence of 1,000 new viruses from a single infected cell. By the 1950s, virologists had established these fundamental aspects of viral behavior. But the development of virology did not stop there. For example, science knew almost nothing about the variety of ways in which viruses cause disease in us. In the 1950s, scientists were unaware of how viruses evade immune defenses or their rapid evolution compared to other life forms. They also did not know that a virus had transferred from chimpanzees and other apes to humans, becoming what we now recognize as HIV, one of the deadliest viruses in history. They could not have predicted that by 2020, a new virus, COVID-19, would emerge and lead to a global pandemic, though ultimately it would be overcome. At that time, the significance of viruses extended beyond their role in causing disease. Scientists were yet to understand that viruses play a crucial part in the genetic diversity of all life on Earth. The study of viruses, their emergence, and their impact is still a complex and evolving field. In this video, you will discover what viruses truly are, why the first living organism might have been a virus, and the intriguing fact that we are 2% Neanderthal and 8% virus. You'll also explore how we differentiate between living and non-living things, and consider the provocative idea that humanity itself could be viewed as a disease. If you want to support my work, please like, comment, and subscribe. Everything around us, from the tree outside your window to the fly buzzing by, your pet cat or yourself, is categorized within a clear framework. The distinction between the animate and inanimate is often straightforward. For example, a crystal, while it grows over time, is inanimate, and fire, despite its movement, is also considered inanimate. In contrast, bacteria, worms, birds, and dogs are alive because they possess the essential properties of life, including being composed of cells and engaging in metabolic processes involving proteins, fats, and carbohydrates. Viruses challenge the traditional understanding of life. Biologists have varied perspectives on what viruses truly are. Sir Peter Medawar, an eminent immunologist and Nobel laureate, once humorously described a virus as bad news covered in proteins. Generally, viruses are defined as non-cellular pathogens that live inside host cells. They are minute entities composed of a protein shell and nucleic acid. While viruses differ in characteristics such as the shape of their envelope, the presence of surface projections, and the structure of their genetic material, they all share a basic repetitive structure. Unlike other pathogens like bacteria or fungi, viruses do not exhibit all the characteristics of life. They lack metabolism, do not grow or change on their own, and cannot reproduce independently. Instead, they must hijack a host cell's machinery to replicate and produce new viruses. According to traditional definitions of life, viruses are considered non-living because they do not meet the criteria of metabolism, cellular structure, irritability, and self-regulation. Despite this, viruses contain nucleic acids, which are used to synthesize proteins. Although this protein synthesis occurs within the host cell's ribosomes, it is fundamentally similar to the process seen in living organisms. Thus, while viruses are not considered truly alive, they are carriers of genetic information and share some functional similarities with living cells. Viruses are a kind of transitional form between living and non-living matter. If we consider viruses to be living, why are they so unlike the rest of life? Why do they have to parasitize healthy cells, thus disturbing the balance? That is, deplete the host organism, and sometimes even kill it. But if we consider viruses to be non-living, why do they reproduce and evolve? There is no clear and simple answer to the question of whether viruses are alive. And it is unlikely to appear until mankind finds an answer to another, probably even much more important question. How did life, and more importantly in the framework of our history, viruses, come to be? The short answer is simple. We don't know. Viruses, as far as we can tell, have been around as long as life itself has existed. Scientists once analyzed the protein sequence of viruses and concluded that viruses may have appeared on our planet independently of life, meaning that during the evolution of the rest of life, viruses continued to appear rather than evolving once from some common ancestor. This, in turn, may explain why viruses are so different from the rest of life. There is no universal virus that can reproduce equally successfully in human cells, bacterial cells, or insect cells. Nor are there species of living things that are not affected by viruses. There are viruses that can infect fungi, plants, animals, and even bacteria. A virus that parasitizes bacteria goes by the name bacteriophage, which means bacteria eater. The main problem for those trying to investigate the origin of the virus itself is that viruses are nothing more than DNA or RNA genome fragments encased in a protein shell. They leave no trace in the paleontological record, and all that remains to study their past are modern viruses and their genomes. By examining genomes and analyzing their similarities and differences, biologists are uncovering evolutionary connections between various viruses and identifying their most ancient features. Despite their incredible diversity, all viruses share a common trait: they are parasitic. Viruses cannot perform independent metabolism. They rely entirely on the biochemical processes of their host cells. They lack the systems needed to generate ATP, the primary energy currency of biology, which powers movement and thought. This dependency makes viruses intracellular parasites, unable to survive on their own. This raises a compelling question. How could viruses have emerged separately from other forms of life if their existence is so tightly bound to cellular life? To address this, scientists have proposed three main hypotheses about the origins of viruses. The Vagrancy Hypothesis. The fact is that our DNA is a very ancient information archive, a library, the filling of which has been changing for 1 billion years. In the course of evolution, mutations appear in DNA. The configuration of genes changes. Just as in a library there are books that no one borrows, in DNA there are sections that encode nothing. There are two types of regions in any DNA: informative exons, nucleotides that code for some protein, and uninformative introns, which code for nothing. Exons are, within our library metaphor, 1984 or Harry Potter, and introns are yellowed recipe books. Introns are usually cut out of RNA, a copy of DNA, so that they don't then interfere with protein synthesis. This process is called splicing. The Vagrancy Hypothesis suggests that some of the introns may have left the genome, escaped from it. Back in the 20th 100 years, science encountered jumping genes, transposons, a large group of introns that are also called mobile genetic elements. Jumping genes can calmly change their place of residence in the genome, moving from one to another. Moreover, there are quite a lot of such genes. They account for up to 47% of the insect genome, up to 69% of the mammalian genome, and up to 90% of the plant genome. Theoretically, some transposons could escape from cells and start the evolution of viruses. Is there a downside to this hypothesis? Of course. First of all, it is completely unclear how the escaped introns got a capsid, that is their protein shell. It is impossible to imagine that the capsid appeared by chance, even the simplest one, not to mention its more complex variants, such as the recent coronavirus, for example. The second hypothesis is the degradation or simplification hypothesis. According to this hypothesis, all viruses were once ordinary cells, which in the course of evolution moved to a parasitic intracellular lifestyle and lost absolutely everything except their own genome and a small protein body. The hypothesis suggests that back in the era of unicellularity, there were large numbers of parasitic cells living inside other cells. Such cohabitation of two cells does not seem unrealistic in general. There is ample evidence that, for example, mitochondria, intracellular energy generators, and plant photosynthesizing plastids, plant solar cells, were once nothing more than bacteria that got into a large cell and were left to live in it. This is, of course, about the process of endosymbiosis. It is quite possible that the ancestors of viruses were such cohabitants, but for some reason, after many generations, they degenerated and became militant nomads moving from cell to cell. It is also known that representatives of some modern cellular parasites, such as Rickets C, are relatives of mitochondria. They share a common ancestor. Indeed, they can live and multiply inside the host cell. What also adds interest is that the size of Rickets C is 0.1 micrometers, which generally corresponds to the size of large viruses. And viruses, as you already know, are very small. To make it clearer to you, the size ratio of a virus to a human being is like a human being to the Earth. Rickets C lost their autonomy a long time ago, although unlike viruses, they have their own metabolism, which does not depend directly on the metabolism of the host cell. Does the degradation hypothesis have any disadvantages? Yes, it can't explain such a strong degradation clearly enough. The same Rickets C and other intracellular parasites retain the features of a full-fledged living cell. And if we take their even more degraded relatives, mitochondria, we can notice that, despite the complete non-self-sufficiency in mitochondria still perfectly guessed once quite independent cell, mitochondria divide themselves, and their division does not depend on the division of the host cell. They still have a genome one way or another. Other than that, they have an active metabolism. There is another hypothesis that scientists are unlikely to ever disprove or confirm. Let's call this hypothesis the shadow hypothesis. According to this hypothesis, viruses appeared before the appearance of the most ancient, common to all life forms last universal ancestor, or simultaneously with it. That is, the development of viruses ran parallel to the development of standard cellular-like cells. Viruses are thought to have originated in the primordial soup, emerging chaotically from nucleotides, simple enzymes, and amino acids. According to this hypothesis, viruses represent one of evolution's attempts at creating life. Although these attempts may have failed in terms of complexity, they have been remarkably successful in terms of distribution and abundance. For instance, scientists studying seawater discovered that each liter contains up to 10 billion viruses. After doing the calculations, the researchers concluded that there are about that many viruses in the ocean. This amounts to 10 nonillion viruses, an almost incomprehensibly vast number. To put it in perspective, there are 100 billion times more viruses in the ocean than there are grains of sand on all the beaches of the world. If you were to weigh all the viruses in the ocean, their combined weight would be equivalent to 75 million blue whales. To put that in perspective, there are about 10,000 blue whales left on Earth. If you lined up all the viruses from the ocean in a chain, it would stretch 5,700,000 light years. Despite the challenges in verifying the shadow hypothesis, it might ultimately prove to be correct. Confirming this emerging and promising hypothesis would revolutionize our limited understanding of the origin of life. It suggests that viruses could have existed long before the separation of bacteria, archaea, and eukaryotes, which is why viruses affect all three domains of life. RNA is believed to have been the first life molecules, with its self-replication and evolution leading to the development of all life on Earth. The earliest viruses might have started as highly aggressive RNA molecules, which later acquired genes to encode protein codes. The evolution of viruses is even more complex than the evolution of all life forms combined. It's possible that all three hypotheses about their origin have merit in different ways. These intracellular parasites may have emerged independently through various processes. For example, Giant DNA viruses might have originated from the degradation of ancient cells, while some RNA-containing retroviruses could have evolved separately. Alternatively, the emergence of viruses might be attributed to an unknown, undiscovered mechanism. Besides the origin of viruses, we are interested in the question, can a virus be considered alive? What is life in general? The difficulty with the question of whether viruses are alive is that viruses are not devoid of signs of life. They are much more alive than something composed of inorganic matter, like a rock or a crystal. It would be an oversimplification to categorize all microorganisms smaller than viruses as non-living, and those larger as living. Organic molecules, though essential building blocks of life, do not themselves constitute life. Viruses occupy a liminal space between life and non-life, or might embody both states simultaneously. The question, what is life, was put in the title of a very famous book by physicist Erwin Schrödinger in 1944, and prompted a whole generation of physicists to engage not only in physics, but also in thinking about this fundamental question. Life adheres to the principles of thermodynamics and energy conservation. Living cells are marked by negative entropy, which means they maintain organized structures despite the natural tendency toward disorder. Entropy, often seen as a measure of disorder, can be likened to a messy apartment that becomes more disordered if left uncleaned. Only by putting in effort, essentially working against entropy, can you restore order and cleanliness. Similarly, life uses energy obtained through nutrition to impose order on its surroundings. Schrödinger's inquiries were focused on the principles governing life itself rather than its origins. NASA, in its quest to identify life beyond Earth, has developed a clearer definition. Gerald Joyce, a researcher formerly at the Salk Institute, significantly contributed to this definition by creating self-replicating RNA in vitro that could mutate and evolve, an attempt to simulate the origins of life. It is quite possible that these experiments inspired the space agency to formulate such a clear definition of life. It goes like this. Life is a self-replicating system that contains genetic information and can evolve. Viruses, based on NASA's definition, are alive. In this context, viruses can be compared to apples. An apple lying on a table cannot duplicate itself and turn into two apples. The same is true of a virus. An apple needs soil to become an apple. Accordingly, the question is, are apples alive? Just as an apple needs soil, a virus needs a nutrient environment to replicate and produce offspring. Bacteria are commonly thought of as living organisms. They have the ability to divide and thus reproduce themselves. Most importantly, they synthesize protein. Protein synthesis is considered an important boundary marker separating living and non-living things. Bacteria need nutrients from the outside world, meaning they are not completely independent microorganisms. There is no biological equivalent of a perpetual motion machine, no mechanism that can function without external energy sources. In the absence of sunlight and nutrients, energy must come from other sources, such as chemical reactions. For example, life at the bottom of the ocean thrives around hydrothermal vents, known as black smokers, where chemical energy is abundant. This environment is thought to be where life originally emerged. Recently discovered giant viruses, known as mimiviruses, have components essential for protein synthesis, akin to biological robots performing various functions. These giant viruses closely resemble bacteria and even host smaller viruses that replicate within them. The discovery of mimiviruses has challenged classical virologists, as these viruses defy traditional definitions and concepts about viruses. The 2013 discovery of mimiviruses was discussed in the journal Nature, where it was suggested that these giant viruses might be placed at the base of the tree of life. However, the scientists who discovered them recognized that neither cells nor mimiviruses could have been present at the origin of life due to their large size. Therefore, early viruses likely existed before the advent of cellular life, and modern viruses, as parasites, evolved later. They are dependent on cells. A parasite virus can transfer its functions to its host, and leave the host with fewer genes than if it were on its own or had to survive outside the host cell. Evolution not only goes from simple structures to complex structures, it can also go in the opposite direction. Complex structures can become simpler, can lose genes, delegate their functions, and become specialized. Depending on environmental conditions, abilities can be gained or lost. So how do viruses interact with the host cell? It all depends on the host. There are host cells that do not have a nucleus, such as bacteria and archaea, prokaryotes, and cells with nuclei are eukaryotes. They are made up of insects, fungi, plants, and animals. The life cycle of a virus can be described as follows. Viruses enter a cell to infect it, then remain in the cell, integrate, replicate, and can destroy the cell. For instance, herpes viruses can persist in neurons for years, while many plant viruses remain in the cell indefinitely, never acquiring a shell, remaining inactive, and replicating alongside the plant cell. Additionally, retroviruses and some DNA viruses integrate into the host cell's DNA, introducing new genes. This integration can sometimes have genotoxic or mutagenic effects, potentially damaging the host cell. Viruses are capable of destroying the host cell, releasing their many thousands of offspring. Moreover, this is often in response to stress. In such situations, for example, herpes viruses crawl out of their shelter and end up on our lips. But why do viruses often just kill, destroy their hosts? Are they interested in our survival? What we know for sure is that humanity has long been a kind of evolutionary ark, protecting some forms of life from extinction. Around 10,000 years ago, following the agricultural revolution, the populations of staple plants like millet, barley, wheat, rice, and corn began to grow exponentially. Concurrently, humans started domesticating animals, leading to the proliferation of species such as sheep, cows, goats, chickens, horses, dogs, and cats. These domesticated animals became integral to human societies, and their numbers grew in tandem with human populations. The key factor for a gene's success is the number of individuals carrying it. Life forms that humans can utilize, like cats and wheat, benefit from this association. However, viruses are not included in this human ark. Particularly dangerous viruses are fought by mankind. Although if you think about it that way, diseases that are not very deadly, pharmaceutical companies play into their hands. The same situation is supposed to be true for viruses. Viruses that are too deadly should not survive. After all, they simply exterminate their hosts, burning the soil for themselves. If most cells disappear, there are so few of them left that viruses will simply not find a useful cell. So when host cells are scarce, viruses adapt to new types of hosts. We are talking about such a dangerous phenomenon as zoonosis, due to which the human body is infected with completely new animal viruses. Virus typical for some Chinese bats, in the course of very rapid mutations begins to infect humans. That's what happened with the Ebola virus, for example. It was transmitted to humans from bats. The same thing happened with the SARS virus. The genetic and evolutionary connection of all life, including viruses, is much tighter than you might think. Now, I'd like to introduce a bold statement. Humanity is a virus. The biological evidence will surprise you. The clever can learn much from the enemy. Aristophanes. Elysia chlorotica, a striking leaf-shaped sea slug native to the salt marshes of the east coast of the United States, is a fascinating example of a plant-animal hybrid. Its unique green coloration hints at its remarkable ability to photosynthesize. This sea slug accomplishes this by incorporating chloroplasts, organelles responsible for photosynthesis, from the algae it consumes into specialized cells in its digestive tract. These cells then proliferate, spreading the chloroplasts throughout the slug's body and forming a dense layer just beneath its skin. Once this layer is established, Elysia chlorotica stops eating altogether and derives all its energy from the photosynthesis of the chloroplasts. During its evolution, genes necessary for producing proteins were transferred from the algae's DNA to the slug's DNA, a process facilitated by viruses that inhabit the slug's tissues and cell nuclei. These retroviruses, though ancient and not fully understood, play a crucial role by integrating their DNA into the slug's genome and enabling the transfer of genetic material from the algae. This unique blend of plant and animal characteristics is further complicated by the slug's life cycle. At the end of its life, as it emerges from hibernation to lay eggs, the retroviruses activate, leading to the death of the host slug. This phenomenon isn't accidental. In every Elysia chlorotica that completes its life cycle, retroviruses are found to be actively multiplying, causing pathological changes in the tissues indicative of a viral assault. The crucial point is that these pathogenic viruses are not introduced from outside sources. Rather, they originate internally. The conclusion is that the sea slug is ultimately killed by its own retroviruses, which played a vital role in enabling it to harness solar energy. This represents an evolutionary strategy where the symbiotic viruses orchestrate the programmed death of their host species. This case exemplifies aggressive symbiosis, a potent evolutionary mechanism where viruses not only facilitate symbiosis but also lead to the host's demise. Viruses are remarkable evolutionary agents, excelling in mutation. They mutate at rates thousands of times faster than bacteria, and bacteria mutate at rates thousands of times faster than humans. This rapid mutation rate is crucial in medicine as it contributes to the development of drug resistance in viruses and bacteria. The challenge in treating diseases like AIDS and tuberculosis stems from this rapid mutation, necessitating a combination of drugs to combat the swiftly evolving pathogens. Viruses have profoundly influenced the evolution of life on Earth, contributing significantly to the diversity of organisms we see today. For humans, viruses have played an especially pivotal role. In fact, a virus closely related to HIV was instrumental in the evolutionary process that led to the emergence of humans. I understand that these assertions might seem incredible or unsettling, and you might find them confusing. So let's get to the bottom of it. In February 2001, the full sequencing of the human genome was publicly revealed, marking a monumental milestone in biology and one of the greatest scientific achievements of the 21st century. However, for those outside the scientific community, the significance of this breakthrough might seem obscured by complex terminology. Our genome is relatively modest in size, encompassing about 20,000 genes. Each protein is encoded by specific regions of DNA known as exons, which are interspersed with non-coding sections called introns. Additionally, regulatory regions of DNA govern how and when genes are activated. The human genome sequencing revealed some astonishing findings. Most striking was the realization that only about 1.5% of our genome is dedicated to what defines us as warm-blooded vertebrates. In contrast, nearly 9% of our genome is composed of endogenous retroviruses. This presents a paradox. While the core components of our genome are relatively small, the vast majority of our genetic material is made up of remnants of ancient viral infections. This paradox reflects the legacy of billions of years of evolution, highlighting the profound impact that viruses have had on shaping our genetic heritage. At the time of the human genome's decoding, scientists were not fully aware of the implications of their findings. For decades, there had been a reluctance to view viruses as symbiotic entities, leading to various misunderstandings and miscommunications. To advance our understanding, it was crucial to address these issues. Every moment, living organisms experience a myriad of interactions with viruses, involving complex exchanges of genetic material between different branches of the evolutionary tree. This includes interactions from the simplest bacteria and archaea to more complex organisms like fish, birds, ants, monkeys, and humans. While many of these viral interactions occur in peripheral tissues and do not significantly impact evolutionary processes, some can occur in vital organs and directly influence evolution. Viruses, with their ability to integrate into and manipulate their host's genome, can sometimes invade germ cells, sperm or eggs, and be passed on to future generations. This form of genetic integration opens up significant evolutionary possibilities, as it allows for horizontal gene transfer, where genes are shared across generations in a non-traditional manner. This horizontal gene transfer can lead to the emergence of new species not through simple vertical branching, as suggested by neo-Darwinism, but through the merging of genomes via symbiosis. This concept highlights a broader and more dynamic view of evolution, where viruses play a crucial role in shaping genetic diversity and driving evolutionary change. It is difficult to imagine a more stringent and effective selection than culling by means of an active symbiotic virus. From a traditional Darwinian perspective, any invasion of the host genome by a virus can be interpreted as a selfish genetic action for the survival of that virus, and it may well be that natural selection would act in a largely individualized way for the virus and the host. However, as we have seen with AIDS, in the midst of a severe epidemic, we can observe selection operating largely at the level of symbiosis. During the active spread of the epidemic, it is logical to expect the unpleasant consequences of aggressive symbiosis and the destruction of a significant part of the host population. But this stage may not last very long. The culling leads to the creation of a new partnership between the virus and the host. The remaining members of the species reproduce again, and these remaining members who interact with the virus gain an evolutionary advantage. The philosophers of antiquity, Japanese and Hindus, believe that everything in the world is composed of 4 basic elements: earth, water, fire, and air. And if the inclusion of water, earth, and air in the basic elements is understandable, for these are the constituents of visible matter, fire is an exception. Fire is a fundamentally different and maybe even hostile type of matter. It is destructive. It brings disasters. It is the consequence of wars and natural disasters. It erupts from volcanoes. It spreads over a city during a battle. At first glance, it seems to bring only suffering and death. So why did ancient cultures include it among the basic elements of nature? Perhaps because of its alchemical role in the creation of the new. After all, fire extracts metal from ore, extracts copper, iron, and gold. The properties of fire are dualistic. It can destroy, but it can also serve to create. From an evolutionary point of view, viruses have the same duality. There are viruses that we are infected with today. There are also viruses that we don't get infected with, but our distant ancestors did. In the same way that the genome of modern humans has 2% of Neanderthal genes, we have genes from ancient viruses. The human genome is 8% made up of fossil viruses. We see sequences in it that are characteristic of viruses, for example in the type of proteins they encode. However, unlike the viral genome, such sequences are not harmful to the body. The sheer number of mutations through which these genes have occurred prevents viral particles from reproducing. These viral genes can move around our genome. The discovery of gene migration within a chromosome is one of the greatest discoveries in biology, which belongs to the eminent American scientist Barbara McClintock. Barbara conducted her experiments on corn, which has gone down in the history of genetics as the Mendeleev pea. In 1948, she was the first to discover that there are genes, transposons, that seem to jump around the chromosome. In the 20th century, there was strong discrimination against women in science, and the discovery was not believed at the time. However, in the end, in 1983, Barbara received the Nobel Prize for it. Subsequently, it turned out that there are quite a lot of such elements that jump around the genome. The endogenous viruses inside of us have also turned out to be among these jumping elements. The idea that human genes might be of viral origin is, by virtue of its provocativeness, a bit too glib. We tend to think of our genes as our complete identity. The fact that bacteria acquire a significant proportion of their DNA from viruses raises uncomfortable questions. If bacteria evolved from viruses, is it really possible to detect such kinship in other life forms as well? Are we all just Frankenstein's viral monsters? At first, this problem could be separated from our own existence and viewed solely as a problem of microbes. The presence of their viral genes seemed a mere fluke of lower life forms. But today we can no longer rest on that. Now, when we look into the human genome, we also see viruses. An endogenous retrovirus affected the common ancestor of all placental mammals, who lived over 100 million years ago. Today, this virus slumbers in the genes of armadillos, elephants, manatees, and humans. Doomed to live in the host, the endogenous retrovirus can retain the ability to produce copies of its DNA that are reinserted into the host genome. Over the millions of years that endogenous viruses have invaded our genes, they have accumulated an incredible number of them. Each of us carries nearly 100,000 fragments of viral DNA in our genome, which is, as you already know, up to 8% of our entire DNA. To appreciate just how big that is, let's remember that the genes that code for proteins, the enzymes that do virtually all the work in our bodies, are only 20,000, and that's only 1.5% of DNA. About 100 to 150 million years ago, an ancestral mammal was hit by an endogenous retrovirus. It helped form the first synthesized protein that eventually allowed the placenta to form. Over millions of years, this original placental mammal gave rise to many lines of descendants. They continued to be infected by endogenous retroviruses. In various lineages of mammals, rodents, ungulates, and primates, some viral proteins were replaced by others. At the most intimate moment of new human life budding, viruses are essential for our survival. This suggests that we cannot exist in isolation from the rest of life, and even from viruses. There is only a constant intermingling of DNA. It is to viruses that we owe the existence of the placenta. There are endogenous viruses that regulate our immune system. They fight modern viral infections. There are sensors in our body that respond to the presence of pathogenic viruses. The immune system works quite slowly. When first infected, it takes about a week for the immune system to start fighting the infection. Therefore, evolution has created a system that alerts the body before it figures out what has gotten in, similar to how fire alarms go off in buildings without understanding the cause of the fire, and turn on water to put out a fire. We have a very similar system in our body. It's also inherited from ancient viruses. As you probably know, we are in a very close relationship, literally in symbiosis with bacteria. We have 10 times more bacteria in our bodies than our own cells. We are more a part of some bacteriological system than the bacteria are a part of ours. Bacteriophages, literally the police for bacteria, play a role in regulating the life of our bacteria. Bacteriophages keep our bacteria in check, regulating their numbers when the balance is upset. In particular, much of the defense against sex infections in women comes from fermented milk bacteria. Our intestines, stomach maintain an acidic environment that allows us to destroy most pathogens that enter us with food. It is the bacteria teaming up with viruses that provide internal balance. As we look into life, we discover an infinite number of cause and effect relationships that make us think about the unified nature of all living matter, both human and virus. If something looks like a fish, swims like a fish, and smells like a fish, it is probably a fish. It's roughly the same with humanity. From the outside, our cities look like tumors on the body of the planet. Our species has spread throughout the body and is engaged in activities that are obviously destroying the balance of the body. Today, the Earth is almost completely developed and controlled. Mankind is looking at neighboring planets and will probably try to reach them. Mars is a prime candidate. It's pretty cold there, and you know how they want to warm it up. Just drop some warming nukes. No other organism has ever spread at this rate and over this kind of territory. We lived in Africa for the first 150,000 to 200,000 years. Then the climate changed and humanity began to spread across the planet. We got to the Middle East 70,000 years ago. Then we got to Asia. 15,000 to 20,000 years ago, someone ended up in the Americas. Very quickly, mankind occupied both American continents. Some even swam to Australia. Today, there are no places left where our feet have not set foot. Sometimes you look at some small islands in the Pacific Ocean, find out that someone lives there, and ask yourself, how did you get here in the first place? In fact, the answer is simple. There were unoccupied territories, and they decided to occupy them, just like deserts or permafrost areas, where people live for some reason. Even after a nuclear war, some number of people may survive. Some will band together, some will build a dictatorship, and some will build a cannibal society. No matter how devastating potential climate or political crisis may seem to us, humanity will endure and gradually recover. Our bodies cannot tolerate high temperatures, just like viruses. That's how the human body defends itself against infection. The body raises its body temperature, and often that's enough. As with any virus, humanity destroys other life forms or uses them to sustain itself. Some species are exterminated completely. Others manage to put in the red book, but it does not help much. Viruses are found that are faithful to their calling, and sooner or later they get their kicks. If now there is a surviving representative of prehistoric fauna, some dinosaur or even more ancient reptile, there will probably be hundreds of thousands of people who would not mind hunting. For the first 300,000 years, humanity evolved and grew stronger. Subsequently, those who jumped on the Ark of Homo sapiens and became human food resources managed to maintain their population. Wheat fields stretched to the horizon and poultry farms were built one after another. Gradually, technological superiority elevated man to the top of the food chain, leaving other animals no chance. But it wasn't enough. At a certain point, the planet itself was in the crosshairs. Only humans are capable of changing the parameters of the planet so dramatically. Changing the parameters of the Earth is usually unintentional on the part of man. Man simply digs up minerals, builds factories and energy infrastructure, expands the boundaries of cities. From the outside, the places where minerals are extracted look like rotting wounds on the body of the planet. In addition, humans are polluting the world's oceans, taking away access to clean and safe water for themselves and other life. And water, as we know, is the main component of life. The process of globalization is multifaceted. As we began to move resources, both natural and human, we began to tackle the entire planet. We were interested in every corner of it, every patch of forest, water or air, regardless of who or what had lived there before our arrival. Resources, benefits, food, useful applications. Man needed everything at once. We learned to make chemicals by extracting them from the world around us, and to synthesize them ourselves. Thanks to science, we have a coordinate system that includes a detailed description of everything around us from our galaxy to our own bodies. We learned to compete with each other, to sell, to improve, and to utilize the art of alleviating suffering in war and for our own survival. The era of suffering was completed by Edward Anthony Jenner in the late 18th century. He was an English physician and scientist, but he went down in history as a pioneer of vaccination. In 1796, Jenner injected some pus from the blisters of a cow into the arm of 8-year-old James Phipps infected with cowpox. He then proved that Phipps was immune to smallpox thanks to the vaccine he had made. The world community of scientists was skeptical, but Jenner's successful experiments were published in 1798, and gradually the world became convinced that he was right. Jenner coined the term vaccine from the word vacca, which is Latin for cow. Along with other discoveries, industrialization, and new advances in military medicine, our march across the planet, as well as our population growth, will begin to steadily accelerate. Our battle against disease will harden. Human life will be declared the supreme value, and the right to it will be included in the list of basic rights and freedoms of all states, established by the constitutions. We will begin to systematically destroy everything that prevents us from living and reproducing, competing with populations of our own species in speed and efficiency of propagation. We have come a long way from being part of the diversity of species and the evolutionary struggle for supremacy, where adaptability was the most important element, to endless breeding and resource consumption, where we began to remake the world around us to suit ourselves. Homo genus has acquired a new trait. It exalted itself above the rest of the living and non-living world, becoming an infectious agent in their relation and acquiring features characteristic of another domain. Viruses. Any cell is an ecosystem in its own right. When a virus invades a cell, it begins to use it, pulling all possible resources from it, turning the ecosystem into a factory for the production of its own kind. From the outside, the Earth strikingly resembles such a cell infected with a virus. However, I advise you not to be deceived, not to drown in dark gloom. Viral nature is natural for Homo sapiens, but human nature is available. Each of us should not forget that the meaning of life is to resist entropy, to be able to go against the current of universal destruction. The path of creation is available to each of us, the path of not mindless multiplication and capture, but of structuring influence, of creating a new combination, an additional possibility, of opening a new horizon, a horizon that can be explored. Thank you for watching. Special thanks to sponsors. If you want to support my channel, put a like and subscribe.